Well, welcome everyone to the Science for Citrus Health webinar today on managing Asian citrus psyllid and citrus growth using particle films. We're excited that you all are here. Just to get started, there's a link here to our website. We'll put that in the chat box as well if you would like to go check it out and get some resources and also find out what other stuff is happening. How you can participate today in the webinar. If you have questions throughout the presentation, please put them in Zoom's Q&A that's at the bottom of the ribbon bar. And at the end, there'll be time for a Q&A panel, and we'll read those questions off to the speakers and address them. If you have any technical issues or comments or discussion, you can use Zoom's chat feature for that, but please don't ask questions of the speaker in the chat box because they might get missed. And also, if you want just us panelists to see your chat or comment or the entire group, make sure you change who you're sending it to. There's a little drop down menu. We do have continuing education units available today. From these three organizations. The California Department of Pesticide Regulation, CPR, has 1 hour of other CEUs approved. There's a course code here. You will have to answer questions, poll questions, little quiz at the end of the presentation today. And then after today's event, you'll be emailed a final exam that you'll have to then take and receive a passing score of 70% or better. That instructions and all of this information will be emailed to you again at the end of today's event. So look out for that email and instructions if you need credits. And then for Texas and Florida, we also have 1 hour of CEUs approved from both of those states. And so if you need those course credits, send us an email after today's event with your first name, last name, and license number, and we will send you back a certificate at today's event. So with that, I would like to introduce our speaker for today is Dr. Christopher Vincent. He is an assistant professor of horticultural science at the University of Florida. He'll kick us off. So Christopher, take it away.
Hi, everybody. As you already know, my name is Christopher Vincent, and I'm trying to move my presentation into presentation mode. Okay. Please, somebody let me know if that didn't work. Yeah, the title of my talk today, and actually I should say our talk today because this is in coordination with Dr. Monique Rivera at UC Riverside. And she'll be presenting some of the results later on in the presentation. The title is Managing Asian Citrus Psyllid and Citrus Growth Using Particle Films. So I want to go over the main points of what we'll talk about today before we start to talk about them. And yeah, so first we're going to talk about shading and leaf temperature in citrus physiology and why that's important and how it could affect growth and yield. And we'll discuss results using white kaolin, which is a natural kaolin color. It's available. It's effective in Asian citrus psyllid management. It delays HLD infection in areas where there is HLD transmission actively occurring. It improves growth. It has some limitations in terms of efficacy during high flush periods, and it has a challenge with rain fastness. Red kaolin, which is essentially just the white kaolin with a dye added, can be produced in the tank. It is more effective in managing Asian citrus psyllid and HLD. It offers a greater improvement in growth. We think it improves water use efficiency. Still a question mark there. There's more rain fast. So it overcomes a few of the challenges that we see with white kaolin. Some of those challenges, of course, with the rain fastness, that's a bigger problem for us than it is for y'all. So the thermos here is actually to cue up a joke about Texas A&M, but I won't tell the joke. But basic idea is that these treatments have to do with maintaining the optimum temperature for citrus trees. So citrus trees are actually originally forest trees, and not forest trees like overstory oaks or pines. They are understory forest trees. Right here, this is a picture of myself with Myrtle, a Nirvan biological scientist, and postdoctoral associate in my lab. And we're actually right there, we were performing a study. The juice wasn't part of the study, the plants behind it were in natural areas. So these are grapefruit trees behind us growing in the shade of oaks and a few other larger species. So that's sort of the natural habitat of citrus to grow understory or in gaps sort of on the edge of forested areas. So then we have a question. Are citrus sun plants or shade plants? They have a number of really interesting attributes. One is they have really thick cuticles, so that helps them not lose water too fast. They also have photosynthetic stems, and this matters in that it changes how the canopy as a whole responds to light dynamics versus how an individual leaf does. So for a long time, we've known that citrus trees have a low saturation point. What that means is that at a low level of light, they achieve their maximum level of photosynthesis, and more light doesn't lead to more photosynthesis. On the right-hand side here, you see the long dashed line. That's from an individual leaf. And you see it reaches saturation there somewhere around 600 to 700 micromoles per meter squared per second. So what is 600 micromoles per meter squared per second? That's a little less than 1/3 of full sun during the summer months. So midday full sun during the summer months. So that leaf doesn't photosynthesize a whole lot more if you begin to add more light all the way up to full sun. However, when we measure photosynthesis on a whole shoot level, which is more similar to how a tree canopy is growing, we see that it doesn't saturate until we get very close to full sun, more than 1500 micromoles. So what that indicates to us is that on a whole shoot level, in terms of the plant's ability to take that light and turn it into photosynthesis, or into sugars, I should say, through photosynthesis, they act more like a sun tree, which, that begs the question: how is it that studies using shade have increased citrus growth and yield? And why do we think of them as shade plants or partial shade plants? We'll get there in just a second. So this is leaf temperature data from a study that we have in the field in Florida. This data was collected in June, and the red line represents a fully sun-exposed leaf, and you see there that it peaks the leaf temperature peaks at about 113 degrees Fahrenheit. So that is not a temperature we ever see in Florida. The temperature outside was about 96 degrees Fahrenheit. Now, the green line represents a leaf that was on a tree that was in about 50% shade. And you see that its peak was about 98 degrees Fahrenheit, just slightly over what the air temperature was. So what that means is that high light leads to really high temperatures in citrus leaves. And the reason it does that is because they don't have the capacity that some plants have to cool through transpiration. So just like us, plants allow water to evaporate from their surfaces in order to cool. And the more you can do that, if the climate is dry enough, the more you cool. But citrus trees don't do that very well. They have quite low conductance. And we'll get there in just a second. This is what happens under different shade conditions to the leaf water potential. So anything below, say, like minus 1.5 would be like, this is a healthy leaf, not water stressed at all. But these values down here below 1.5 with the 0% shade, that represents a water stress leaf. Now, this is not under dry conditions. This is Florida in the summer with irrigation. So what's happening with these leaves is that high light and the heating of the leaf is causing it to lose water faster than the tree can move that water into the leaf. And so because of that, and because the tree needs to prevent the leaves from drying out, it lowers stomatal conductance. So this is a plot of stomatal conductance over the course of a single sunny day. And what we see here is that most of the conductance happens when over the course of about 3 to 4 hours of the day. Now, why is conductance important? It's important because that's how carbon dioxide gets in so that the plant can do photosynthesis. So the plant will open up its stomata to allow carbon dioxide in, but when it does that, it loses water too fast. And so high light, high leaf temperatures means that it's losing water too fast. It has to close those stomata to prevent it from drying out. And then as it closes its stomata, leaf temperature starts to climb. As the water comes back in, it reopens those stomata to try to reestablish sort of normal function, right? Lower temperature and higher photosynthesis. But then it again loses too much water. And so it recloses its stomata and it spends a lot of time below this 0.1 number that doesn't have any intrinsic meaning, but we use it as kind of like a rule of thumb to say anything below this is very low conductance and anything above it for a citrus tree is acceptable. What that indicates is that the majority of the photosynthesis that's occurring during the day is happening in these little periods where I've drawn these little blue boxes around. So that's somewhere between 3 and 4 hours in the course of that day. The rest of the time, the leaf is more or less shut down. And that is induced by that combination of high leaf temperature, high light, that could be reduced by giving that plant a little bit of shade. So that's what brings us to particle films. Particle films are just suspensions in water of particles that are then sprayed on the leaf to form a film on the surface of the leaf. What they do is they reflect light, and through that reflection of light, you get shading of the leaf that particle film is on, and you also get some redistribution of light into the canopy. So for instance, maybe this leaf might be getting too much, and if you have a particle film on this leaf, some of that light will get reflected away, and some of it will go deeper into the canopy where the leaves weren't getting enough light and lead to greater growth and photosynthesis. They're also used to reduce sunburn. The picture on the bottom right-hand side is a picture of some mandarins that I took in the Central Valley in California in, I believe it was July of 2019. And that's just the kaolin particle film that had been sprayed on that you see a flush coming out after the particle film application. The rest of the pictures are sweet orange trees in Florida. One of the questions that I get a lot, and so I always include this little picture, this black and white micrograph at the bottom. I get asked a lot whether these materials block stomata. And so I include this picture, which came from Jim Syverson, who was a physiologist at the center for a long time, basically to demonstrate kind of how these particles tend to distribute themselves around stomata. That image there is a picture of a stomate, and then those little dandruff-looking things. Those are the particles of surround. So particle films can be made with many different materials. They can be made of titanium or aluminum, all sorts of things. But the most common is kaolin clay. The reason being that you can mine it straight out of the ground and it comes out white. And so that's the cheapest material to acquire. We are experimenting with including red dyes, and there are a few reasons for that that I'll get into. And well, actually, So this is a part of the dye preparation process originally before we figured out how to do it in the tank. The reason we're interested in adding this red dye is that there was some background work that was done here at this research center that indicated that that particle films would help reduce the arrival of Asian citrus psyllid, which spreads citrus greening on a tree, but that if you dyed it a different color, you could actually enhance that effect. And they found that bright red color was the most effective of the various colors that they tried, including all sorts, purple, blue, yellow, green, everything, dark red, bright red. So originally we had to mix this with some solvents and things to get the dye to adhere to the kaolin, and so we had a long process that involved several cement mixers set up in our lab. Thankfully, we don't have to use the solvent anymore. We have a material that's available on the market that can be mixed and creates very similar color without the involved process or the dangerous chemicals. So here's how they look when applied to young orange trees out in the field. Occasionally, we get some grasshoppers painted as well. And so just a look of the pink color that results from adding the red dye to the kaolin in the field, and then you can see close up what it looks like on fruit, both the white and the red. The picture in the top left, that's a jug of color back. It's just a mulch colorant that has iron oxide, which is what gives it its red color, and some sort of adhesive that helps it to stick. So what does the leaf look like when it's applied in terms of the two things that I think we should pay attention to? One is the degree to which the color attracts or repels psyllids, or I shouldn't say repels, attracts or does not attract psyllids. And then also we need to pay attention to photosynthesis, right? That matters for the plant response to these treatments matters. So what we see here is this is the reflectance of a leaf at different wavelengths with the different treatments. So I apologize, the red is the control, not the red isn't the red treatment. Bad graph making here, but the red line you see below has less reflectance. That's the control. So that's a leaf with no particle film. And then you see the green line is with the red kaolin, and the blue line is with the white kaolin. Overall, what you see is that the white kaolin has much higher reflectance across most of the spectrum. And for reference here, I've included a visible spectrum. So that's the part that we can see, that little bar that goes from purple to red. And the wavelengths that are known to attract psyllids are designated with these arrows, right? So that's yellow, green, and then just before ultraviolet. And then actually even getting into UV. The ratios of those things matter in ways that I don't know have been quantified enough for us to be able to make a calculation on this. But what we do see is that in all three of those regions, you see less reflectance with the red than you do on the white. So that's for comparison of the red to the white. The two wavelengths regions that are most important for photosynthesis are blue and red. In fact, that's why you see those decreases in reflectance from the leaf in those areas. That's because the leaf is absorbing those colors of light. So you can see that the red also reduces blue in particular in terms of what it's reflecting back. And then red to far red ratio affects how stomata work and some flowering signals and some other aspects of plant responses. So based on this, we hypothesized that the red would be effective against Asian citrus psyllid, but not so much in terms of positive effects on trees. We thought that the white would be horticulturally superior. So we started with a new planting. We planted all new trees and treated them that same day that we planted. These are all Hamlin sweet orange on Harsky rootstock, and we tested those trees for roughly once a quarter over the course of the following 2 years. Tested them for sea lice infection, so that's the bacterium that causes HLB. And what we saw was that quite quickly, actually, by the second sampling, we already had major levels of infection. So that's like less than a year, but major levels of infection in foliar insecticide treatment and in an untreated control. And we had much lower infection in the white and the red kaolin. Interestingly enough, we did not detect any HLB positive trees in the red treatment until well over a year. So it's July of 2018 after planting. And you can see that that date lines up with the foliar insecticide treatment reaching almost 3/4 infection rate. So 75% infection rate versus 15 at that time. However, over time, in our conditions, these treatments still do become infected. And we'll get into kind of how that might happen. And so by July of 2019, so that's a little over 2 years after planting, we had 100% positive infection, which is pretty standard here in Florida. So this is what the Asian citrusillid populations looked like on these trees in response to the treatments. Basically, what we see is that in the first year, we found very few Asian citrusillid on kaolin treated trees, both the red and the white. Just a few here and there. It wasn't common to see them until we got to March of 2018. So that's just shy of a year after planting. And those trees produced a very large spring flush that grew out so quickly that it covered the mature trees, the mature foliage that had the kaolin on it. So the new leaves,
Yes, so I'm decently familiar with this data, so I'm going to hop right in until Christopher's audio comes back. So the figure that we're looking at right now is a long-term study that Christopher did in Florida on young, freshly planted trees. So what they were comparing here is a foliar insecticide treatment in pink, and then the red kaolin and untreated control and the white kaolin. So those are these lines with sort of a band on them. So what you're looking at is psyllids per tree. So they actually, when the trees were young, were looking at all of the psyllids completely over the entire tree. And then in later years, we're actually doing similar to what we do, where we'll check flush points.
Can you hear me now?
So, okay, Christopher's back. So I'm going to hand it back over to him. And then I'll be back in a sec, guys, when he's done with his portion.
I'm very sorry. I bumped my microphone and it has a loose connection, I guess. Thanks for jumping in there, Monique. Okay, so where was I? These are the Asian citrusillid numbers. All observable Asian citrusillid per tree. Yes, it did take a lot of work. No, I didn't do most of the work myself. The people whose pictures you see down here are the ones who did most of the work. And for almost the first year, we had very few Asian citrusillid on the red and white kaolin. That's the gray and the pink dashed bars, until we got to March, where the floral flush just completely covered up the treatment. And new flush, the particle films don't adhere well to new flush. And so we couldn't reapply until after those had matured, and that's where you see those numbers declining again. You do see some higher numbers the next year,
in the floral flush. That's right during March of 2019. And you also see some during the summer of 2019 when those trees have gotten quite large. The untreated control typically initially had much higher psyllid populations, where you can see that the insecticide is suppressing those populations, but over time you start seeing more psyllids per tree in the insecticide. And I'll get into why in 2 more slides. So when we take those numbers per plant and sum them up over the whole study, what we see is about 450 Asian citrus psyllids per plant in the foliar insecticide treatment, a large but smaller number in the untreated control, a much reduced number, less than 100 in white kaolin, and then a little bit further reduction from the red kaolin. And we also went back because we thought the growth differences were beginning to accumulate between the treatments. We went back and we did, over the course of about 6 weeks, we did every other week, we looked at just new flushes and quantified the adults and the nymphs that we observed on those new flushes. And what we see is a very similar pattern to what we saw in the overall data from the whole plants, where we see a reduction from foliar insecticide to the untreated control, and then a further reduction from white kaolin, and then a further reduction from that in the red kaolin. So I mentioned some differences in growth. This is the trunk cross-sectional area. So that's the growth of trunk girth, which is one of our better proxies for total growth that we can do without destroying the tree. And what we see over time is that pretty early on, the red kaolin begins to exceed the other treatments. Over time, the white kaolin also begins to separate from the untreated control and the foliar insecticide. One thing we do see is that also relatively early, the foliar insecticide treatment, that's that solid line with the circles, grows more than untreated control. We think this is actually the underlying reason behind why that treatment had more psyllids, is just because there's a lot more tree there to be attractive to psyllids. So psyllids can find that larger tree with more foliage more easily. So even though it was protecting the tree, it also got more psyllids when the insecticide wasn't there to suppress it. The kaolin treatments, on the other hand, have a little bit longer residual because they remain on the surface of the leaf for longer. One of the things that we do, because when you get 2 trees, they're not exactly alike, and so then when they grow, what we do is we deal with relative growth rate. So that means that how much the tree grew relative to where it started. And what we see is that the foliar insecticide and the untreated control pretty similar. Red kaolin and white kaolin had much greater relative growth rates of trunk cross-sectional area than the other 2 treatments. When we look at the relative growth rate of the trees as when we consider whether they were infected or not infected with HLB, what we see is that uninfected trees had much higher growth rates than infected trees. This, of course, is not surprising, but I want you to look at the scale at which that effect occurs. In other words, the effect of growth reduction of HLB in terms of trunk growth is similar. The reduction is similar to the amount of increase that you get from applying red and white kaolin. Then in terms of what we got totally, this is data that was taken about 2 years after planting. You can kind of see, just as an illustration, this tree, which was the white kaolin, is a good deal taller than the untreated tree that's right next to it. And you see a red kaolin and a white kaolin tree next to each other. Similar size. I'll show you another example in just a second. But what we see is that canopy volume of the red kaolin is greater than all of the others. The height is slightly larger than the white kaolin, but much significantly greater than the other 2 control treatments. And then in terms of leaf area index, that's kind of like a measure of canopy density. It's an important measure of plant health and estimator of future growth. We see that white kaolin and red kaolin are similar to each other in terms of canopy density. So the red kaolin has a larger canopy, but that canopy is similarly dense to the white kaolin. So here's just a kind of an illustration. This is CloudPoint data that came from a company that came in and did some LiDAR scans of our trees. So they scanned every single tree in our 2.5 acres. And these are the median trees in each treatment. So in other words, this no insecticide tree that was half of the no insecticide trees are greater in canopy volume, and half of them are smaller in canopy volume, and the same with each of those treatments. So this is the middle one of all of those, and you can see the kind of size and volume differences that we're talking about. I just kind of outlined them in blue just so that, because the cloud points weren't I didn't feel they were clear enough. But so you can see how the red kaolin is slightly larger than the white, which is significantly larger than foliar insecticide, which is much larger than no insecticide. And of course, the thing that we all care about the most is yield. And so this is yield from 2018, 19, and 20 in that planting. So obviously it starts out very low because it was a new planting planted in 2017. So the first year, nothing. Second year, very small, just a few fruits really. And we just gathered them because we just wanted to get an idea of what their sort of carbohydrate health status was and get an idea of how they might grow in the future. And so you see that each year they grow more and more within each treatment, but also you see pretty big differences in terms of between treatments. So we see that the 2 kaolin treatments, even though the red kaolin, the mean is higher, but they're not statistically different in terms of how much yield they produce. And then those two are much higher than the foliar insecticide and the untreated control, which yields almost nothing. And just this past harvest season, we measured soluble solids. And what we found is that the trees that had kaolin applied, the fruits were about 1 point higher in terms of soluble solids. Now, of course, these Brix numbers are atrocious. Nobody wants to eat this fruit. But there are a couple of reasons for that. One of them is that this past season in Florida was pretty rough for Brix of juice oranges. And the other is that these are still young trees, which tend to produce lower Brix in the first place. So I'm not touting this as excellent soluble solids numbers. However, 1 point increase is meaningful. So one of the things that we wondered about was what's leading to these growth differences that we're observing, both between the red kaolin and the white, as well as in the between kaolin-treated plants and untreated plants. And one of the things that we're considering was that citrus plants in Florida often undergo sort of transient water deficits when the conditions are dry, because of our very sandy soils that drain really fast. And so that typically happens like late fall or late spring, where we tend to have still relatively warm temperatures, but not much rainfall. And so a student of mine, Juan Pablo, he did this study where he looked at water use in these potted trees when they were treated with the different treatments. And so what he did was he re-watered each pot every day, weighed how much water it had lost, and then replaced that water, except that he imposed a very mild water deficit. So a little bit of a drought treatment, not too extreme, just mild, where he only replaced up to about maybe like 75% of what they were losing. And what he found was that if you expose them to this mild deficit, they would reduce their total water loss. Not too surprising. So The gray bar on the right-hand side, that's the well-watered treatment. And then the dark gray bar on the left-hand side, that's the water deficit treatment with no particle film. But then what he found is when you added particle films to these trees that had that were being exposed to a water deficit was further reduction in water use, at least with the red, not so much with the white. So then he looked at leaf water potential. So this is kind of our measure of whether the tree is undergoing some sort of a biologically significant water stress, right? And what he found was that both particle film treatments essentially made the leaves just like a tree that had full water, whereas the only one that had a reduced leaf water potential was that one that was being exposed to the water deficit. So with that, we're seeing that these particle films are impacting leaf water status. And so we're thinking that if you think back to that graph that I showed where the stomatal conductance is going up and down throughout the day and it's staying very low for part of the day, we're thinking that these treatments actually allow the plant to maintain a higher stomatal conductance for more of the day, and that is essentially the process that allows these trees to grow more with the particle films. I've got a student who is in the process of doing that right now, the same one who produced that stomatal conductance data. Now, another question that we get a fair amount is that now that we have this red stuff that we mix with the white stuff that makes it stick and it can stay on through all this rain, well, is that going to cause a problem when it's time to try to sell these fruit, and are they going to look like they're painted with a bunch of pink paint? And so the Peace River Packing, a local citrus packing company, was gracious enough to let us run a bunch of fruit through their packing house. And these are fruit that we applied massive amounts. So this is a much higher load than what you would expect out in the field from having been applied for pest management or growth purposes. We just really laid it on as thickly as we could onto already harvested fruit. And this is on the bottom is what they looked like when they came out of the packing house. So you can see very clean, not really observable residue. So I was glad to see that because we don't want to find a solution that creates another problem. And so with that, it's time to take these treatments to California and see how they do. So I'll hand it over to Monique.
Okay, so I think you should see this full screen. So I just want to start out by first thanking all of the California citrus growers and the Citrus Research Board for funding this project and the collaboration between Christopher and I. It has been a complete pleasure and super fun to bring this technology to California and have a look at how it works here. So secondly, I'd like to thank everyone that's on the call. I see people from all over the state, and I really hope to see you guys in person really soon. However, if you do like webinars, please let us know in the follow-up survey, because we want to best serve you and transmit the information in the manner that you guys prefer. And lastly, I have a small caveat for my organic growers that are on the call. The dye used in this study is not currently organic approved. We did try to run it through CCOF, and that was not successful. So that is still a question that's remaining in the development of this technology. I do believe that this tech would be an amazing tool for organic growers in California. Okay, so the photo that you're looking at is on Limoneira's property. So we thank them for that collaboration, and my technician, Joanne O'Sullivan, who collects all the data in Ventura County. And then also Associates Insectary and Brett Chandler, who applied this trial for us. We are greatly appreciative. So this is a drone photo of these small, they're not fully bearing yet, lemon trees. And I'm going to give you a presentation on the data. So our pressure in these groves is not super high, not like you would see in, say, San Diego County at this point. And these are very well managed groves in terms of insecticide and keeping the plants as clean as possible. So essentially what our results are showing is that the way this works in terms of ACP and their attraction to the plant is mirroring what they found in Florida. So very conveniently, the pink stands out the best on these figures. And so you can see in almost every instance, the trees with the pink treated kaolin have the lowest ACP pressure. Now, there are some points at which you'll see pink peaking, and what's not included on this figure that next time I present this, I will make sure to include is when we had the applications, because sometimes it was hard to get the treatments applied in the timing that they need to be, because it was looking like these small trees were growing out at the rate of about every 6 weeks. However, what we found in the data is that with the pink kaolin, there is less pressure when we get the grow out of the flush. So that's very important, because with the white kaolin, this flush becomes very attractive. And this is in part due to things Christopher explained, which is that in terms of light reflectance, that white reflectance is essentially going to reflect all colors and make that flush that's grown out this, basically a flag waving saying ACP, come find us, where the red background really helps distract their finding ability. So we did this also in a second location in Ventura, and these are results here as well. You can see overall number of nymphs for certain. You will always see that the pink is lower, and that's because, again, that finding ability. But when you look at number of adults, say per tap sample, you might find adults there, but it doesn't seem that they are laying their eggs in the pink kaolin, or even necessarily in the white kaolin. But tap samples can be a little confusing to interpret, because there may be other ACP on the plant that aren't caught in the particular tap sample. Number of adults, which is the top figure on the right-hand side, is a visual inspection, trying to find the adults visually on flush points. And similarly, in mirroring the nymphs, you would see the most on the untreated control, with white and pink suppressing greatly the adult visual numbers. But these numbers are still quite low. So we also did this work in Riverside on navel oranges. This was applied by Washburn and Sons. I do see Irene on the call, so thank you guys so much as usual for all your collaboration. And this, we had higher pressure, as you can see with the lines here. So let's start out with the figure all the way on the left-hand side. This is number of nymphs per flush. And again, what's missing here are our treatment dates, because as I mentioned before, when the treatment will wear off, that's when we would start to regain the pressure, and it is hard to keep this applied. However, these trees were fully grown. And what we found is they were not as well managed as the trees under Limoneira. So we felt that that was keeping the flush from growing as fast as it normally would. So for a large tree, these treatments, essentially, it looked like they were lasting for about a full 2 months. So that's a pretty decent amount of coverage and residual time, certainly more residual than any insecticide is going to provide for ACP. So again, let's look at our nymph numbers. Here you'll see the nymphs peak a little bit in the pink. And I really do think that based on, if I had the treatments dates on here, you would see that that is related to the treatment wearing off. However, when the nymph numbers start to build a lot, you see the huge peak in the untreated control getting to really high numbers. So now moving over to tap samples. Tap samples consistently overall had the most in the untreated control, but you do see some peaks in the pink. And I really think this is due to the flushing patterns in those older trees that weren't flushing as strongly. The grove's not as well maintained, again, as Limoneira maintains their trees. So average number of adults visually. So this is a visual inspection of the flush when you inspect for the nymphs on the flush points. Essentially, the untreated control is where we would find the peaks, but the pink treatments kept the visual adult numbers down the lowest of all of the treatments. So CRISPR was kind enough to provide this preliminary tree growth information. And it's particularly important for the young trees in Ventura, where they're trying to get those trees as quick as possible into full production. So what we're looking at here is the canopy volume as measured by Joanne painstakingly with a measuring tape, the lengthwise, and basically holding it at the side of the canopy and trying to figure out what those sizes look like in terms of where the leaves span. And so essentially what we found in Limco Del Mar, and again, I do think this relates to our ability to get stuff retreated. We're seeing that as the statistics break out, that the white treatment in Limco Del Mar had the best growth. However, if you flip that and go to Orchard Farm, essentially you're finding that the pink treatment is greatly overtaking the untreated control and the white. But in both instances, aside from in Limco Del Mar, the white and the pink, that kaolin treatment is enhancing the growth of the canopy volume. So in conclusion, particle films, we do believe that they have great potential for ACP management and hopefully in California to reduce any sort of potential disease pressure. Overall, between the two states, the red dyed kaolin is coming out as a better treatment. And when we use this color back, which is pictured here on the bottom right, this creates even more rain fastness. And so that's why I was mentioning that those treatments on those older trees lasted for 2 months. We could really clearly see them on the trees when we went and scouted. So despite that, and folks in the desert know this for sure because they do their whitewashing every year to protect their plants from the extreme high temperatures, these treatments, regardless of the color, do increase growth and yield. So when it comes to making sure to prevent HLB infection, what it seems like from Christopher's data is that the red or the pink treatment is superior if it gets applied before the tree becomes truly infected with HLB. And that happens very quickly in Florida, something we're trying to avoid in California. So in Florida also, the treatments with kaolin both maintain higher growth with and without the infection. And as per Christopher's research in Florida, the particle films improve leaf water status and mitigate water deficits. This is going to be increasingly more important in California, as we all know. Would be great to look at this as it compares to irrigation rates in California, because as everyone in the citrus industry recognizes, in particular in the Valley, there are continual water wars going on, and any way that we can create more efficient irrigation is going to benefit everyone. So what we found, and also this has been done by Beth Grafton Cardwell before she retires, kaolin does wash off in the packing house, but the packing house needs to have the correct infrastructure to be able to wash off the kaolin. Not all packing houses, as I learned very quickly in Ventura County, have the infrastructure to use for, say, water to include in the washing process. So what's going on currently and what the continued work is going on in terms of what Christopher's doing. So what happens when you apply kaolin to a mature planting after canopy closure? Does it have the same benefits? That's a key question that needs to be answered. Effects on fruit drop and fruit size. And then in California, we really need to get an organic certified dye so that we can get this to our organic growers who do not have the insecticide selection that the conventional growers do. We want to prevent and mitigate insecticide resistance. We cannot just depend on pyrethrins. So with that, in conjunction with Dr. Christopher Benson, I'd like to thank all of you, like I did at the beginning. Citrus Research Board and CRDF have funded this work, and I think that this is an exciting new technology that has really interesting applications. So with that, I will conclude the webinar and we can start to answer some of your questions.
Before we get into that, thank you, Monique. This is great. I do want to launch this poll for DPR questions.
The first question that we have in the poll is, what factor most affects the efficacy of particle films against Asian citrusillid? Option 1 is sunlight, option 2 is humidity, option 3 is rainfall, and option 4 is earthquakes. Number 2 is particle films reduce Asian citrus psyllids by camouflaging them so the psyllids don't perceive a citrus tree. True or false? The last question, question number 3, is particle films reduce growth of trees by plugging their stomata. True or false? Yeah, number 1, the correct answer is rainfall. So that washing off of the treatment, that's going to inhibit the efficacy of that treatment greatly. So number 2, particle films reduce Asian citrus psyllids by camouflaging them so that the psyllids don't perceive a citrus tree. That is true. Number 3, particle films reduce growth of trees by plugging their stomata. The correct answer was false there.
Okay, so especially for number 1, for those of you who put sunlight, please make note that that is not the correct answer. The right answer is rainfall. All right, so then with that, I guess we can go into some questions. We have quite a few in the Q&A box, so I'm just going to start from the top if that works. I believe this one was for Christopher. Is the dye something special? Otherwise, how would you change the rain fastness?
Yeah, so the dye itself is not something special. The dye, what actually creates the color is iron oxide, and so that I would argue is not something special. However, it comes in a mixture that has proprietary ingredients that cause it to adhere really well, and I have not been able to figure out what those are exactly, but they work really well if you're trying to not have to reapply every 3 or 4 days in Florida. Going back to that rainfall issue. So it's not the dye itself, it's not the colorant itself that causes it to change the rain fastness, but this product as a complete package does.
Okay, great, thanks. The next question, what about other materials, anti-transparent, like, Willet, Perf, anti-stress? There's a few others in there. How to remove films from fruit?
So I have not compared these products with any of those products. What I would suggest is that the way these work is helpful in as much as it reduces the transpirational load. So if the product, which I think like VaporGuard works by inducing stomatal closure, it would not have a positive effect in that way. But if the product uses some sort of light reflectance or shading, where it decreases the demand of the environment for the water that's inside the leaf, then the effects could be similar.
Great, thank you. Is red kaolin commercially available and approved in California?
So I can answer that question. So as far as the regulatory environment, I've gotten approval from DPR that this is not a problem to apply it as we did in this trial. I didn't have to do any sort of special use or anything. In terms of your specific situation and your region, you might want to check with your ag commissioner or other certifier before going ahead to apply this. But my understanding in conversation from DPR that there was no limitation on this adding of dye to the kaolin because it is not changing the nature of the use of the kaolin product.
Okay. Great, thank you. Somebody else is asking, where can growers obtain the kaolin? What is the mixture formula?
So it's just basic Surround or the generics of Surround kaolin. And then I think later on in our study, or maybe we can send out a follow-up, we can give you the ratios of mixture.
Perfect. It says, why wasn't calcium carbonate added to the study? Was it was market red used to try and dye the kaolin?
Yeah, so calcium carbonate was not used in this study, and we had a couple of reasons for that. One is that it's not as reflective as surround. Another is that it's not chemically inert, and so it can, unlike surround, it can have effects on other chemistries. And so we thought that surround or kaolin in general was a better bet because it's inert and is more reflective. We did test a lot of different dyes, and the biggest problem with, I'm not familiar with all of the specific brands, but the biggest problem with all of the spray marker type of dyes is that they're extremely water soluble. So as soon as they're exposed to any water, they are dissolved into that solution, and they will come off in the dew or in the rain or the irrigation or whatever. So they don't bind to the kaolin. So we didn't use those for that reason.
Okay, great. Christopher, as well, do you think you would get the same results with fully grown trees?
So that's a big question for us. As you saw in the slide with the sort of next steps, we're currently doing that. So we have a couple of trials in production settings with larger trees. We've got one with about 30-year-old trees and one with about 10-year-old trees. We suspect that there may be differences between the results that we got and and what happens when you have a larger tree with a denser canopy, with the dense canopy being the most important aspect of that. My current hypothesis is that white will actually be more beneficial in terms of growth for fully grown trees. The reason being that dense canopy can benefit more from that reflectance working its way back into the denser parts that are otherwise more shaded, whereas the red won't do that quite as much. But there's a lot of back and forth there. In Florida in particular, anytime you're working with mature trees, you are working with HLB affected trees as well. And so there are interactions moving different ways that could even mean that we're talking about a different effect in Florida versus California in terms of how they impact mature trees.
Great, thank you. Monique, are the treatments only done once?
No, they are not. So we did the best that we could in Ventura to keep up with the flush grow out on those trees. So they were reapplied every 6 to 8 weeks. At some points we were hitting closer to 1 month when we would get strong grow out, but this was all in Ventura County. We had to get in line with the other treatments for those trees. So sometimes we could be operating on a bit of delay. So they didn't restructure their entire pest management program for our kaolin applications, because this is a commercial grove. And in addition, in Riverside, this was the same thing, but we found that flush grow out was less intense on the larger trees. So like I said, 8 weeks on the larger trees was sufficient.
Okay, great. Are there any adjuncts used for better disposition of the kaolin?
For a while, we were using a spreader sticker called SKH to improve rain fastness. We still use that with the white, but we don't use it with the red anymore because the sticking effect is no longer needed. But and as far as like better spray pattern, we don't. In fact, we did find that some adjuvants actually caused larger droplet formation and had caused problems. And that's something that has the specific chemistries of those adjuvants can interact with the chemistry of the leaf. So we started off in our earlier project testing the adjuvants that were recommended by apple growers who regularly used surround, and those did not work. Some of those did not work for the application of surround to citrus trees because the chemistry is must be somehow different, cause large droplet formation and stuff. So now we do use that one spreader sticker. It's the best one of all the ones that we've tried. It doesn't get us where we want to be in terms of Florida. But it's probably not a huge deal for California under most circumstances.
Awesome. Also for Chris, those dark markings on the oranges, is that normal in Florida? What causes this if it isn't the red dye?
Yeah, so actually those were pretty nice looking oranges for Florida.
Okay.
Yeah, so the russeting, that's from mite damage. And because these are juice oranges, growers don't take any measures to prevent aesthetic blemishes. They don't have any effect on juice quality. So typically, if you see a citrus truck driving by during the harvest season in Florida, the colors that you see are like brown, black, green, yellow, a little bit of yellow, a little bit of orange, mostly brown. And that's because mite and other insect damage, as well as rusting from wind damage. But growers in California, or excuse me, in Florida, don't typically take measures to prevent those for juice oranges, just because it's not worth the cost.
Great, thank you. What was the rate of kaolin that you guys were using?
So we go with an equivalent of, so the typical spray rate in Florida is about 125 gallons per acre. And we go with 50 pounds, of surround per 125 gallons of water. But then with the red, because it shades more, to achieve the same level of shading, what we do in Florida now, and sometimes I forget details. Money, please correct me if I'm wrong. I think we did this in California too, where we use 35 pounds, the equivalent of 35 pounds per acre of surround, 35 pounds per acre, or 35 pounds per 125 gallons. And we go with the dye, we use 2.5 fluid ounces per pound of kaolin.
Okay, wonderful. Let's see. I've heard some talk about the current colorant being difficult to remove or possibly staining some rinds. Is there any information you have about that concern?
So they ran it, that's going to really vary by packing house technology, as I mentioned. Yeah. So that's my only way to address that. I think that I know for sure CCH here in Southern California does have the technology to wash stuff off, because I had to ask a bunch of packing houses. So that's good for organic growers that put their fruit through that packing house. However, my understanding in Ventura County was, because it's Brett asking this question, is that I wasn't aware of a packing house in the county that had that technology. So that would be something that we would need to do in a follow-up study if we can get the fruit properly treated. And I think for this study, right, Christopher, you just direct sprayed the fruit and then ran it through the packing house?
Yeah, we really laid it on there for that one. But yeah, so what you see there is we harvested the fruit and then sprayed it and then took it to the packing house. So yeah, but the packing houses here, as far as I understand, they all typically use a water detergent wash. So they just ran it through the regular process and didn't have any trouble.
All right, good question. Another question here in the chat box: how effective is kaolin in extreme heat, such as here in Southern California last September?
So is this question in the chat?
Yes.
I'm trying to find it. How does it hold up in the heat? Is that the question?
How effective is it in the heat? Yeah.
So it's very effective. If we're talking about just the white surround, in the sense that in the desert in Imperial County, they regularly use this to protect their trees. So essentially think of it as like a sunscreen for the trees. We do the same thing with zinc oxide and titanium dioxide on our bodies.
Okay. Let's see. Cheryl wants to know, have you ever tried regular spreader sticker?
We've tried a lot of different spreader stickers. I'm not sure which one a regular one would be, but most of them did not have a really strong effect on rain fastness. I wish I had, excuse me, I wish I had included some pictures of this, because we hooked up a giant shower head and sprayed them, and then we measured how much came off. But anyway, most of them didn't help at all. SKH was the only one that helped noticeably. There were some others that maybe helped a little bit, but I think that most of the spreader stickers work based on their impacts on the surface tension of water and that sort of thing. And so when you're talking about spreading a water-soluble chemical around, it's very different from spreading around a particle that, relative to the size of an organic chemical, is very large. So most of the ones that we've tried, and I don't have a list on me, didn't work very well.
Okay, great. What was the foliar insecticide that you used in the trial?
I don't have the list on me, but we used the rotation of about 12 different chemistries that are typical of growers in Florida, that they use with these, or should use with these types of rotations to manage Asian citrus psyllid in particular.
So that's right, people on the call, that's true. They are spraying that much there and to try to keep the psyllids under control. That is not the case in California. We have cooler weather and we can time to our flush patterns, because HLB hasn't completely decimated our collective flush pattern, so that we can still be on the same page when that's happening and treat proactively.
Okay, somebody wants to know, does this affect re-entry period or harvest time at all?
It should not. So this is not, there's no toxicity here. And reentry is usually due to trying to reduce exposure to a toxic compound. And then harvest time would be as normal, as long as your packing house would be able to remove the materials.
And then can this affect quarter elimination?
Yeah, I'm not sure about this question. I don't know what they meant by that.
Yeah, I don't know.
It says quarter elimination, and I'm not sure I even know what that is. Christopher, do you know?
I was wondering the same thing.
All right, well, if Daryl wants to clarify, maybe he can type another question in for us. I'll leave it in there for now. Aaron asks, it definitely, oh, this isn't a question. Okay. Could we use iron oxide with the kaolin? I think it might be something that could be OMRI, maybe. We
might have a proposal being considered right now that says that we want to do just that. Yes, hypothetically.
Okay. Can you mix insecticides?
So I'm not really sure what this person is referring to. So Daryl, if you could type what you're referring to here. Maybe you're asking if you can mix insecticides with the kaolin. I would not advise that, because we do not understand how the kaolin itself will interact with the AI. And if you want to get the effect of the AI, you don't want to disrupt that.
Yeah. Let's see. Do you anticipate any iron toxicity problems with multiple applications? You just kind of briefly touched on that, but.
We don't. for a couple of reasons. One is that we have trees that we've been doing this for several years with and not had a problem. The other is that this is not water soluble iron. These are large particles that don't break down at any significant level, at least. So we don't anticipate that much iron gets in. I've had people ask, well, maybe this could count like our foliar application of iron, and I don't think this is getting into the leaf.
Okay, great. And the last question I have right now, but feel free to keep typing them in if you have more. Did the organic certifier indicate on what grounds dye certification was rejected? Is the manufacturer developing an organic formulation?
So I'll take this question. The organic certifier did indicate on what grounds the certification was rejected, and that was because there is, I think, like 0.025 of some proprietary compound that the dye manufacturer would not reveal, and they kind of made it clear that it was of synthetic origin. So to refer to your second question, is the manufacturer developing an organic formulation? So the original use of that color back dye is to dye mulch. So there is currently no interest on behalf of the manufacturer. This product is not intended for use in citrus to develop an organic formulation. However, between Christopher and I think we have discussed, just trying plain iron oxide as well as beet dye, or kind of extracted and dried colorant from beets. Okay, great. So if anyone else has questions, feel free to type them in. We're all caught up for now. Oh, here comes a few more. Did you put this around out by itself? SKH is just kaolin clay as well.
Yeah, so that's an interesting one. SKH is not it's not kaolinite. It is a different clay mineral, and I do not understand how it improves rain fastness. But we're not talking about the kinds of quantities that you use for applying kaolin. So we're talking about less than 1% of the concentrations. But for whatever reason, the chemistry of that other mineral enhances the rain fastness of the kaolin. That's been observed in other studies too.
Okay, so Daryl has provided a follow-up to what he was. Can this affect quarantine elimination in your area? No, in the sense that if a positive tree is found, you are still going to be involved in the statewide quarantine. So if you did need something else and I'm not answering that properly, Daryl, please feel free to add additional context.
Okay, thanks. Peggy is asking, what happens to kaolin in the soil and does it build up over time? Does it affect the microbial populations at all?
So it can build up in the soil, though the concentrations that you're putting on, take a fair amount of time to build up in any significant level. I don't know about impacts on microbial populations. I would tend to doubt it just because it's considered to be inert. And so it would just sort of exist in its physical effects, but I don't know.
Might there be seasonal advantages using white versus red kaolin in California?
Potentially, yes, potentially, because if you are in the desert and you're dealing with that extreme heat, I would assume or hypothesize that the white is going to be your more effective. You need to reflect as much light as possible off of the trees. So seasonally, it would probably be, you'd have to use white in the desert somewhere. And let's be honest, that could be true of more of the state over time. So, within the scope of ACP, it would mean more so putting it on early in the season when it's not so hot to try to keep down your ACP population. So the RED technology throughout the year in the coastal operations is going to be key. And then Daryl just submitted another question. How can this affect beneficials? So certainly this is going to have an effect on beneficials, but we're not particularly sure of what that is yet, because if this and what other pests it might flare. In our plots, we haven't seen it flare, say scale, which would be the first thing that I would expect. But it certainly will affect beneficials in that I don't think that if it's not really greatly habitable for Asian citrusilla, that it wouldn't necessarily be as great for other insects, would be my cursory hypothesis. However, that's something we'll need to look more into to answer that question with any sort of effectiveness.
And there was one study in Florida before the era of HLB that found increased mite populations with kaolin treated trees. We haven't seen that. We haven't observed that yet in our studies, but I've also heard that anecdotally from growers in other areas that sometimes they see increased mite populations when they're treating with kaolin.
So Aaron wants to know if this data is going to get published or if we get copies of the PowerPoints. Christopher, I don't know, and Monique, if you're willing to let us share that PowerPoint presentation.
I'm totally fine with it.
I am as well, and there is a publication under review, so that's data from Christopher's work in Florida. So I'll be sure to, once that is published, share it with California folks.
Great, thank you. So I've put up the slide with continuing education information once again. Again, you will get an email by 5 p.m. today with all of this information and instructions, along with the link for the DPR final exam. If those of you in California need that credit, it'll come in that email. So the slide is just up for your review. If anyone else has questions, please feel free to ask them. We have a few more minutes. Otherwise, we're happy to end early. And big thank you to Christopher and Monique for this very informative talk. It's been great. Great questions, everybody, all of that. So
yeah, thank you to everyone who attended. We're very excited to share this information.
Yeah, thanks everybody. It's been a pleasure.